Okay, so carbohydrates, or more importantly called sugars, are going to be made out of three specific elements, which is of course in their name, C, H, and O. Okay, we always have a 1 to 2 to 1 ratio. Normally, we have a 1 to 2 to 1 ratio is what I should say. Okay, especially for the hydrogens and the oxygens. Okay, they're always going to be in a 2 to 1 ratio. The carbons are going to be a little different depending on what sugar you're looking at, but for the most part, they will be 1 to 2 to 1. So ribose, glucose, sucrose, those are all very common carbohydrates. And so you can see that all carbohydrates are going to end in OSE. So, basics of the carbohydrate. Okay, any monomer is going to be any one molecule of anything. So, when we're talking about the biomolecules, we're looking at a specific type of molecule. Okay, monosaccharide is talking about one sugar. And so if you look at any one glucose, any one ribose molecule, that is a monosaccharide. Okay, so some of the functions we talked about in class were energy. Okay, you have to use glucose in order to make ATP. It's a short-term storage for our energy. And it's also some other things that you might find in our plants. Okay, their cell wall is actually made out of something called cellulose. Animals' exoskeletons are also made out of cellulose. And so some of those are the things that are going to be found in the animal world where carbohydrates perform a very specific function. So when we look at the monosaccharides, okay, you have three different examples. Okay, this one right here is a pentose because it has this pentagram type shape. These two are hexoses because they have that hexagon type shape. So a monosaccharide is going to be one sugar, one simple sugar. So these are all examples of that. Okay, we also have monosaccharides that are going to be important for other things, and these are some of them, but you don't really have to know them at the moment. A disaccharide, like sucrose, is going to be considered two monosaccharides that are put together, and they are linked by something called a glycosidic bond, which is this O right here in the middle. Okay, they form like this by something called a condensation reaction, which means that you are going to take out a water and replace it with an oxygen in the middle here to bind these two things. So sucrose is actually a disaccharide of a glucose monosaccharide and a fructose monosaccharide put together, and this is what table sugar is. Okay, another example of a disaccharide would be lactose. That's milk sugar. Okay, it's actually made out of glucose and galactose. Maltose is glucose and another glucose attached to it. And again, these are all bonded together by this thing called a glycosidic bond. So when we look at polysaccharides, okay, a polymer is going to be many molecules put together, but specifically if we're talking about carbohydrates, we call it a polysaccharide. And this is a lot of monosaccharides that are going to be bound together again by a glycosidic bond, and they are going to be getting rid of waters here, 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 and here, and all of these are all waters that have left the molecule and are now going to form this oxygen bond. Okay, and this is how we store most of our stuff. All right, we don't want to have just glucose molecules by themselves in our body. Okay, it would be hard for us to keep track of them. So just like when you want to organize a room, you put like things together in a specific place. We take all of our glucose molecules and we join them together by this condensation reaction, putting many of these glucoses together to make something called glycogen. And glycogen is going to be the storage facility, like the storage structure that we put all of our glucose in, and we store it in our liver. Okay, now we store it as glycogen. Plants, however, store it as starch. Okay, starch is again still a polymer. Okay, it's many glucose monomers that are stuck together, but their arrangement is different from that of a glycogen molecule. And so this is how plants store their glucose. Okay, in animals, just like us, we store our glucose as the polymer glycogen. Okay, this is the way that we are going to use it. So it's our storage reserves for when we are doing any type of work that requires energy. We will pull the glycogen from our liver and muscles and use it to do work. Okay, we convert it into ATP. Now, we break down. Okay, so we build up these molecules by something called condensation reaction. Okay, condensation means that water is going to form. So what happens in the condensation reactions is that when I stick these together, okay, 2 hydrogens and an oxygen leave these 2 things to form water, so the water leaves this molecule to form these bonds, And when we're breaking it apart, we do the opposite, okay? We took out water when we made the bonds, this glycosidic linkage. So now what we're going to do is we're going to add back in water. That's why it says plus 7 waters. So we're going to add a water here, add a water here, add a water here, 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 and here. And this is going to break those glycosidic bonds so that I get back my glucose molecules so I can use my glucose to make ATP. But there are other reasons that we would need carbohydrates. Okay, we use glucose as an energy source. Okay, sucrose is going to be used to make things sweeter. We can also break it down into its 2 forms so that we can use it. But there are some things that we just need carbohydrates for, and one of them is going to be structure. Cellulose, which we do not have. Animals do not, well, we as humans don't have it. Okay, we don't make it. Cellulose is a polysaccharide of glucose that is going to be connected in a very straight chain, which means that there are going to be lines of it, lines of parallel glucoses that are going to be bound together. And this gives it a very, very sturdy, rigid structure. And so the plants are going to use this rigid structure in their cell walls to make their cell walls very, very tough. Okay, so what I meant by this unbranched chain means that you have parallel lines of these, okay? Glucoses, glucoses, glucoses. And what they will do is they will bond to each other sideways, but they will also bond to each other down. So this glucose is going to bond to this one's going to bond to that one, and they're going to bond this way. So it makes a lattice. And this is what gives it so, it's very, very, very strong strength. Okay, now if you remember from previously, the starch is actually glucose monomers too, but the cellulose is also glucose monomers. So if they're both made out of cellulose and they're both branched together, how come they're so different? Well, starch is different because all of the oxygens are going to be bound a specific direction. Whereas here, this oxygen's up, this oxygen's down, this one's up, this one's down, that one will be up, that one will be down, up, down. And that's what gives it its rigidity. Okay, so us as animals, we do not break down cellulose. That's why we don't eat a lot of grass. And eating plant matter actually causes us to have very, very weird bowel movements because we don't actually break down cellulose. We don't have the right equipment to be able to do it. However, there are bacteria that are going to be able to do it, and that's why they form a symbiotic relationship between the cows. They live in their stomach, and they help break down the cellulose in return for a place to live and for some of the cellulose material. Okay, another structural form of a carbohydrate is called a chitin. Now, chitin, which looks like chitin, but it's actually called chitin, is another structural polysaccharide, and this is what is going to make up the exoskeletons of many bugs. Okay, it's also going to be what fungus make their cell wall out of. Okay, so bugs are actually going to form an exoskeleton on the back, or on the outside of their bodies, that is made out of chitin, and again, it is very strong. Now, of course, we can crush it, so it's not as strong as like steel, but for a bug, it provides very, very good protection. Okay, and this is just a slide that's going to go over the differences between the four that we talked about. Okay, starch is going to be the way that plants store their energy. Glycogen is the way that we store our energy. Cellulose is going to be found in the cell walls of all plants, and chitin is going to be found in the exoskeleton of arthropods. Now, this was in the first slide for the uses, cell surface markers. So on the outside of the cell membrane, there's going to be lots and lots of things that hang out of it. All right, just like we have radio signals and key passes and locks to enter into the buildings, you have to be able to monitor what's going on the outside of the cell. And so some of these carbohydrates form molecules in a structure that will allow them to monitor what's going on inside and outside of the cell. Okay, it's also going to be important for determining your blood groups, which we will talk about later. So if you look here, this is going to be what the cell membrane looks like. Okay, this is what we're going to go over in the lipids section. These phospholipid bilayers, and in between it, you're going to have these chains that look like hexagons. Okay, here, these green ones. These are going to be chains of carbohydrates that are going to be sticking out from the cell membrane that will be able to bind specific types of chemicals that are trying to get in, monitor how much stuff is going on in the outside of the cell membrane to make sure that the cell is staying within its homeostatic confines. And the last slide is going to be some just vocab that we've gone over in these this PowerPoint, okay? And that's it.